We acknowledge that we are on Treaty 6 territory, the gathering grounds of many diverse First Nation, Métis, and Inuit peoples, whose footsteps have marked this land and whose presence continues to enrich our vibrant community. Hello, and welcome back to Research Recasted, the Knowledge Mobilization podcast. I'm your host, Kelsey Howlett, and today we are joined by Dr. Josh Miller. Josh is an assistant professor in the biology department at MacEwan University. After completing his undergraduate degree at the University of California, Santa Cruz, he went on to intern at the Smithsonian Institute in Washington, D.C. before completing his PhD at the University of Alberta. Afterwards, he held several postdoctoral positions, including at Yale University and with Polar Bears International. As a self-described molecular ecologist and conservation geneticist, Josh uses genetic methods to learn about species in order to help inform conservation techniques. He has applied these methods to a number of species, including bighorn sheep, Galapagos tortoises, pine trees, Arctic Grayling fish, and many others. All right. Hello. Thank you so much for joining us today.
Thank you for having me.
No problem. So I wanted to start just by getting you to tell us a little bit about your background in biology.
Sure. So I was always the kid that loved biology. I basically lived at the aquarium in my hometown. And then in undergraduate, I was a general biology major to give myself kind of freedom to figure out what I wanted to do and explore the different fields of science. But once I found genetics, that's really where I zoned in. And I'm now currently a molecular ecologist and conservation geneticist. So that's been my passion.
Okay. And so a molecular ecologist and a conservation geneticist. Now, I've heard all of those terms, but not quite put together like that. Could you just explain to us a little bit about what that means?
For sure. So molecular ecology is a field where we're using genetic techniques and genetic methods to answer ecological questions. So ecologists can go out and do field surveys, count individuals, learn a lot of information that way. But we can use genetic techniques to see how are those individuals genetically related to one another? How are individuals moving from one area? Is there migration? Is there a decline in genetic diversity over time? And so where conservation comes in is if you see that change in genetic diversity over time, can we put in legal protections, conservation actions to address that?
Okay, cool. So, and I know, so genetic methods to help learn about species and conservation techniques. And I know you've applied these to like a number of different species, like the bighorn sheep and Galapagos tortoises.
Yeah.
Can you tell us a little bit about what those conservation techniques look like in those situations?
For sure. What I love about molecular ecology is that all species have DNA. All life on Earth has DNA. So once we know what techniques to use and what questions we can ask, you can apply it to basically any species. So through my career, I've worked with bighorn sheep, Galapagos tortoises. We'll talk about Arctic Grayling later, I think. Mm-hmm. So for bighorn sheep, which was the focus of my PhD, I asked the question, is there a gene that makes the bighorn sheep have big horns? So people that know bighorn sheep probably have seen them in Bamford Jasper. The males have those really large horns that go around their head. Those are important for the bighorn sheep because males with larger horns tend to gain access to females. And females might choose to mate with male bighorn sheep that have larger horns because maybe they're just more fit or better able to survive. So we wanted to know, is there a gene that makes them have big horns? One, because it's an interesting evolutionary question. How did those sheep evolve big horns? Yeah. But also it's really important from a conservation standpoint because bighorn sheep are a game species. There is a harvest for bighorn sheep, not in our national parks, but other places. And hunters tend to like to harvest sheep with the largest horns. So if there's a gene that makes horns grow large and hunters are removing large horned individuals, you might actually be removing that gene from the species. So it's a bit of a negative feedback loop there. But if we know that, and there is a gene, then you can fold that into conservation strategies. Say, does this population have a high frequency of that gene? And maybe we want to move some individuals from that population to another population where that gene frequency is lower. So that's kind of the genetic technique merging with the conservation action.
That's really interesting. I would have never have thought to ask that question. Like,
Yeah.
is there a gene? And it's also really interesting to see how like specific genes can help inform conservation like that, because that's something I never would have thought about.
That case is actually super interesting too, because in domestic sheep, so sheep we use for wool or meat, there is one gene that controls whether or not those sheep have horns or not and how big the horns are. And so domestic sheep are basically a cousin to bighorn sheep. And so we're like, okay, that seems to be the case in domestic sheep. Is it also the case in bighorn sheep? So there was a bit of a nugget there to start with. Unfortunately for me, it wasn't that simple. There's not just one gene for bighorn sheep. It's a combination of a few genes and the environment. So not a silver bullet there, but still a really cool study system and study question.
Yeah, that would have been my next question was like, did you find the gene?
I wish. That would have been amazing.
But even with like the multiple different genes, can you still use that kind of in the same way?
Absolutely.
In order to like take, okay, so these have like this group of genes, we can still move them over. And then you said the environment plays a big role in that.
Yeah. So you can think about the old adage of nature versus nurture. So nature would be like, what genes are you born with? And then the nurture is what environment are you exposed to at different points in your life, in your life. So for bighorn sheep, the year they're born, if it's a drought year, if it's a really rainy year, those are really important environmental factors that actually influence how large a sheep's horns get later in life. So that's the environment part. But it also matters what genes they have. So if they have the genetic capacity to grow large horns, or if they just have genetic variation that doesn't support that. So there is a push now to build basically a genetic profile rather than just being like one gene. You can build a genetic profile to say, okay, these individuals seem to have the genetic capacity to grow larger horns. And maybe you can use it in the same way I was talking about earlier, where this population seems to have more of the genetic variation associated with large horns. Do we want to move that somewhere else?
That's so fascinating. And okay, so that's bighorn sheep, but you've also applied this to like Galapagos tortoises.
For sure.
Could you tell us a little bit about that?
Yeah. So Galapagos tortoises, people who have heard of them maybe know that Darwin, when he went to the Galapagos Islands, was really influenced by looking at the plants and animals there, including Galapagos tortoises, to form his idea of evolution by natural selection. So you would think that if Darwin had studied them, we'd know everything we could about Galapagos tortoises, but that's not the case. It's still an active debate, even how many species of Galapagos tortoises there are. There's some people that say all the Galapagos tortoises are one species. There's some people that say, no, there's 15 different species sprinkled around the Galapagos Islands. So what I'm trying to help to do is use genomic techniques, so genetic methods to, one, can we say how many genetic species there are? And then two, all of the Galapagos tortoises that are still alive, their numbers have declined significantly since the 1800s, as much as 90%. So there's a big push in the Galapagos Islands to have captive breeding programs to try and build the numbers up and reintroduce them back to the wild. So another big part of the research that I'm contributing to is using genetic techniques to build captive breeding programs, to basically play tortoise matchmaker. Say, these individuals probably should be bred together or encouraged to as much as possible because they're not related. Whereas these two individuals are second cousins or even parent offspring. So don't breed them together.
Wow. So is it like, I know we have like in humans, like families should not breed because it gives way to a bunch of different genetic problems. So it's the same in animals.
Absolutely.
As well.
Absolutely.
That's really interesting. Oh my gosh. Okay. So now we're going to get into your current research, which is around Arctic Grayling, which are a species of fish that are very close to salmon, if I'm remembering correctly.
Yeah, that's exactly right. So Arctic Grayling are a species of fish that we have here in Alberta. They're part of the family Salmonidae. So that includes char, trout, salmon. It's all one big group. And then grayling is a genus within that group of Thymalis. So it's one part of that bigger group that includes salmon.
Okay. And so why why are you studying Arctic Grayling? Like, what makes the species so unique to you?
Yeah. So again, if people have heard of Arctic grayling, it's because they are a sport fish. So fly fishermen in the province like to go out and catch them. They're catch and release here, and we can talk about why later. But they're characterized by having this really big, beautiful dorsal fin that makes for great photos, and they're fun to catch on the line. They're the only member of that genus Thymalis, in North America. But in Eurasia, so Europe and Asia, there's actually 13 different species of this genus Thymalis. So we're interested in, one, knowing, okay, why are there 13 species in Eurasia, but only one here in North America? How did that happen? And two, since they're the only species here, we want to preserve them. We want to make sure that they're around. In the north, there is a harvest, like a subsistence harvest for them. So it's important that they stick around not only for anglers for having fun, but also for subsistence.
Okay. Now, you had mentioned like previously that the ones that we have in the north here are more similar to Siberia than they are to like ones that you would find in southern Alberta. So there's like a lot of diversity. And yeah, I'm just wondering, like, why do you think that is?
Yeah, so there has been a little bit of genetic research done on the species in North America, and that research found that there's two genetic groups, and they're pretty different from one another, estimated to have split from one another about 3 million years ago. And one of those groups is actually more closely related to fish that are still in Siberia, still in Eurasia, and the other seems to be pretty unique to being in North America. Both of those genetic lineages occur in Alberta. It's one of the few places in North America that we've found so far where they co-occur. So it means if a fisherman is fishing in the Hay River, so above high level, they could catch a fish that is more genetically similar to a fish in Russia than if they went to the Freeman River just a little bit west of Edmonton, which is crazy to think about. It's like this fish, you'd think it'd be more closely related to one in high level than one in Siberia.
Yeah, that's so interesting.
Yeah.
Now, because of that, your research is looking to see whether or not these should be classed as completely different subtypes or subspecies, rather than just lumping them all into one sort of one size fits all type thing. Why is that important that we look at them, that we see whether or not they have to be separate or the same?
Yeah. So, I mean, it's the same idea of local conditions would be different. So this fish ranges from Alaska, this fish in North America ranges from Alaska through the territories into Alberta, a little bit of northern BC, and a little bit of northern Saskatchewan. The environment's very, very different across all of those places. But right now, they're saying a grayling is a grayling is a grayling, when really we know Alberta is the furthest south that they come, except for one disjunct or separate population in Montana, but we'll put that aside for right now. Yeah. Alberta is the furthest south as they come, and the climate in Alberta is completely different than the climate in Whitehorse.
Yeah.
So we should probably be managing potentially them differently in Alberta than the Yukon manages this species. But the government needs evidence to do that, right? From their perspective, grayling are doing fine in the territories. We don't need to worry as much about it being fine here in Alberta. That is a Canada-wide policy. There are local management. So there's a management plan for Alberta, that's the management plan for the territories. But on a federal level, they're right now just managed one size fits all.
Interesting. Yeah, that would be important that, you know, different species require different types of management. And you can't just say like, these ones are doing fine, so these must be doing fine. But that brings us back to the fact that the species here is in risk of being endangered and is currently being watched. And I know you had mentioned that they are a game fish and that they're also for subsistence. But why do you think that their population's dying out here?
Yeah, so a recent report found that in Alberta, it's estimated to be like a 60% decline in fish numbers in the province. And that's because they are pretty sensitive to environmental changes. So they like cold, clear, running water to live in. And as soon as there's landscape changes, so development, cut lines, erosion, they are sensitive to that and they would decline in those areas. So there's a lot of monitoring projects to see how our stream temperature is changing. Is there a lot of turbidity, a lot of particulate matter in the rivers, and are fish populations, including grayling, changing because of that? So we're hoping that by bringing genetics to this, again, looking at how many different genetic groups are there on the landscape and where are they, and if they are different from one another, that would be important to know, because if we were planning to maybe move fish from one area to another or try and reintroduce it to an area where they've gone extinct, then we would want to know if this lineage is more adapted to a certain environment. Don't put it in an area where it's not going to survive or not thrive, basically.
Yeah. Okay. Interesting. Now, I know that a lot of these conservation policies and stuff, they are governed by like the government.
Mm-hmm.
As like a conservation geneticist, like what are some of the ways in which we can maybe work towards the conservation of this species?
Yeah. So I think it can really help inform the scale of management actions, right? So is a one-size-fits-all blanket policy acceptable, or does it need to be more targeted? Yeah. Conservation in general is, I think they call it a crisis field. You're always like responding to actions and prior actions, and it's chronically underfunded. So you kind of have to be deciding where you're going to put your conservation dollars. And so if a one-size-fits-all policy works, that's great. That's probably more cost effective than having to do a bunch of smaller policies or smaller actions. But you want it to be more effective. So if those more targeted, perhaps more costly actions are going to be what's needed to keep the species around, then that's justified. So genetic techniques can help us say, where are those populations? Is there migration? Is there a signal of inbreeding, that mating between families? Do we see decline in diversity where the species or the population might be less able to adapt going into the future? And all of those can be folded into management actions, like where do we set harvest areas? Do we have to have harvest quotas? Do we have to close a fishery entirely to allow the numbers of fish to increase? Right now, in a lot of areas, grayling are catch and release. So you can catch them, you can't keep them. But in some areas, it's completely closed to any fishing. So genetically, we can monitor if that complete closure is having an effect on abundance of fish and genetic diversity of those fish in that area.
Wow. So there's a lot of things to think about.
There's always a lot of things.
Yeah. I mean, it doesn't surprise me, like protecting a species in the world, especially with the drastic changes and, people like sporting and fishing and the whole bit. It's a lot to manage, and there's no way that, it's going to be easy to protect these.
And there's always different stakeholders, right?
Yeah.
There's the government, there's the public, there's First Nations, there's researchers. You have to balance all of their ideas, prior experiences, all of that, and thinking about the future as well. So there's lots of moving parts in all of these conservation actions.
Wow. Where are you right now in this research of your Arctic grayling?
For sure. So we just finished, or we're putting the finishing touches on a project where we know that we had those two deeply divergent genetic groups. We wanted to know, are there morphological differences between those groups? Do the fish actually look different from one another, which might signal that they're adapted differently? There's a large body of research in salmon in particular that sometimes you have different shapes of the fish depending on how they live, basically. Like if they're in the open water, they could have maybe a deeper body. And if they live down at the bottom of the ocean, then they would have a wider body, just as an example. Mm-hmm. So we wanted to go out and we actually took photographs of fish to say, what is the shape of this fish? Do we see differences in shape in different areas of the province? And do those differences in shape line up with the genetic lineages that we know about? So we went out and we did that. We took photographs of over 200 fish, wow, as well as tissue samples from those fish. And we did the comparison, and we have the work ongoing where we're looking at those differences. It doesn't seem like it's a slam dunk, unfortunately. It's not like you probably couldn't pick up a fish and say, it's this lineage versus this other one, mm-hmm, but there are differences, statistically significant differences between them. So they might be on the road to becoming completely different, locally adapted fish.
Oh, cool. Well, it sounds like a lot of work, because like 200 fish, that's a lot of fish. And are they from mostly just like here in Alberta?
Yeah, so we focused in Alberta because that's where we are. Yeah. But it's also where those two lineages co-occur. So it was still logistically complicated, but relatively less complicated to go out and do the field work. This was a big collaborative effort between myself at MacEwan, as well as government biologists from the government of Alberta, as well as community partners from Trout Unlimited, though they have recently rebranded. And we actually went out to 6 different places around the province, some close to Edmonton, but we also went up to high level and rented a helicopter to fly almost to the border of the territories to try and get in really remote places to find these fish. So wow, that was an experience. Yeah. Yeah,
no kidding. That sounds so cool. And it's just so fascinating, like where your research is like taking you. Like close to Edmonton, have you been looking at just like lakes and all of that? Is that like where we can find them?
The Freeman River is close to Edmonton. Dismal Creek is close to Edmonton. Those are kind of the most easily accessible that people might have heard of. Yeah. People know where to look for grayling because they are fun to catch, and people like to do that fly fishing. Nice. Yeah.
Okay. So after you've done like this sort of portion of your research, figuring out if the differences in how they look can kind of give you some insight into their their genetic lineage, what's next? Like what comes next?
So I'd really like to take this continent-wide. So there's been a few genetic assessments of Arctic grayling in North America, but they haven't done a genome-wide assessment, so looking at all the DNA in the organism rather than just little bits and pieces of it. And also the eastern extent of the species range, so Saskatchewan, Northwest Territories, they haven't really been assessed at all. So I'm trying really hard to get tissue samples from these, especially eastern edge of the species range, to look at genetic diversity continent-wide, but also genome-wide in the species. Because with advances in technology and being able to do whole genome sequencing, we get much more resolution. So instead of just having a big binary 2 groups, we might find that there's subgroups. And again, we might need to have more conservation actions that are targeted to these different genetic lineages rather than the one size fits all.
Yeah. And then, sorry, I've been thinking in my head. Yeah. Kind of going back to you taking the photos and the tissue samples. So do you have to go out and actually catch these fish? Like, is that how do you go about doing that as a biologist?
As a person that is mainly a lab scientist, I rely on my collaborators. So the government biologists that I worked with, and especially the community partners, they are expert anglers. We had people that had over 20 years of experience, either as government biologists or as just recreational fly fishers. So we worked with them they knew where to look for the fish. They knew how to fish them very efficiently. So we could they could watch in the water. Like we actually did go out and we walked streams and looked for places where the fish were, and they could cast a line right next to it and catch that fish. So it was pretty similar to people that go out and fish recreationally.
Okay.
We were just doing the extra step of going back and taking a photograph and taking a little bit of tissue for genetic analysis.
Yeah. Okay, yeah, that's what I was wondering, if there was going to be any differences in the way you caught them as compared to people who just like go out and catch them. But I mean, it makes sense. Like there's only so many ways you can catch a fish.
Yeah. We also did an extensive ethics process. So there is an animal research ethics board here at MacEwan that we had to submit protocols to, and they helped vet what we were going to do. We did have backup plans if the original fly fishing idea wasn't going to work, but it worked perfectly and we didn't have to do any of our backup plans.
Yeah, I mean, if it's in line with like the catch and release and you're not doing a whole ton of harm to the species. Yeah. That's really interesting. Imagine like I'm fly fishing for science.
It was pretty easy to get volunteers to come out to help us with that.
Oh, I can imagine. Yeah. Okay. Well, I just have one last question for you, and it's kind of a surprise question that we ask all of our guests. And it's, what advice would you give to students, staff, and faculty here at MacEwan? Like any advice at all?
I'm a very collaborative person. I like chit-chatting with people. I like understanding what they do and seeing if it has resonance with what I do and learning about what's out there. So I think being curious and being communicative and having that open mindset to learn about what's going on. I mean, that's why I'm here doing the podcast, is I like spreading information about my research, but then also meeting other people, and collaborative, open mindset as much as you can have.
Yeah. No, I love that collaboration, and especially your comment about curiosity. Because like without that curiosity, you would have never have gone and said, is there one gene that makes bighorn sheep have big horns? That's so fascinating. Josh, thank you so much for being here with us today. Well, that's all we have for today's episode of Research Recasted. If you want to support this podcast, you can visit Research Recasted on your favorite podcast platform to find new episodes every 2 weeks. Also, don't forget to check us out on Instagram at Research Recasted, where you can leave us a like, give us a follow, or send us any follow-up questions you have from today's episode. This has been Research Recasted, a knowledge mobilization podcast brought to you by the Office of Research Services and the Faculty of Fine Arts and Communications at MacEwan University.